All right, so we've gone through the preliminaries on antimicrobials, antibiotics, and others. Um, Talked about terms related to them like uh, uh, narrow spectrum and broad spectrum, um, antibiotics versus synthetics versus semisynthetics, and so forth. We'll review all of that material. Now we're going to go ahead and um, look at uh, the remainder of this chapter. Let's go ahead and look at that. Well, the second third of it, let's say. All right, so yeah, we ended up here with that little concept check. All right, so we talked about the points of vulnerability of bacterial cells, and the one that headed up the list was the cell wall, all right, which is especially vulnerable and suitable as a point of attack because um, we don't have cell walls, number one, and we don't have one of the key components in the cell walls, which is the peptidoglycan. We don't have that. And so we can target this and assume with reasonable assurance that, reasonable confidence that we are not going to be badly affected. At least our own cells won't be. Maybe members of our microflora could be harmed, but um, you can't expect it to be perfect ever. So anyway, so yeah, the drugs that mainly do that are the penicillins and the cephalosporins. They can block the synthesis specifically of the peptidoglycan. So when we say they disrupt cell wall synthesis, what we mean is they're disrupting the synthesis of the peptidoglycan. And if that's weakened, and regardless of whether it's gram positive or gram negative, if that's weakened, that cell is not going to be able to maintain integrity because it's now so weak that if it does take on extra water, if it finds itself in a hypotonic surrounding environment, which is generally the case, it'll take on water and the cell will lyse, all right, which literally means that it explodes, it bursts, all right? It'll go through lysis. Lysis is the root word from which the term lysis arises. All right, and this works best when we're dealing with active, young, growing cells. So um, that would be early on in an infection. All right, so most people are like they feel like they might be getting sick. They just go through this sort of feeling where there's sort of aura. It's like I'm not feeling so good, but I'm okay. I'll keep forging on. And ultimately, then they do get full blown sickness. That's unfortunately most of us. Um, and really, the effectiveness of antibiotics then is going to be kind of limited. So, if you were to do this as optimally as possible, you would take the antibiotic to kill the infection pretty early on. All right, that's when they work best. All right, that of course is a major challenge to be able to do that because it could be that you're really not that sick. You may have something else going on. So it's a challenge. Um, anyway, but that's part of the reason why these drugs have become less effective because people have taken them when they're not supposed to be taking them, and they have more or less put the chemicals out there, and they're in the environment then, and organisms can be killed by them, but not to the fullest extent possible. And you're going to leave behind survivors, and that leads to adaptation ultimately. So anyway, but that's when we want to take them is early on in an infection if we can help it. Um, so there are some penicillins that don't penetrate the outer membrane of gram-negatives all that well. The outer membrane, of course, is composed of some lipid and is hydrophobic and is nonpolar. And to some extent, some of these penicillins, of which, there's many different penicillins now. We've got multiple generations. Some of them don't penetrate gram-negative cell walls all that well, that outer membrane in particular, because of that fact that nonpolar substances are rejected or rejected by polar substances. Anyway, so we've got those. There are some broad spectrum forms that are capable of crossing the cell walls of gram-negative bacteria. And so if someone is infected with one of those, obviously that's what needs to be done. That's what needs to be taken. So up here, this shows kind of the lattice work or the mesh work that is peptidoglycan. When it's fully formed, this is what it looks like. And so we've got these little six-membered structures in here. These are the substances NAM, which is acetylmuramic acid, and then NAG, which is acetylglucosamine. Those are two organic substances, ring-shaped molecules that are major building blocks of peptidoglycan. You can see that they alternate. So you got a NAM and a NAG and a NAM and a NAG. And to join those together, the mortar that goes between the bricks here, if NAMs and NAGs are like bricks, the mortar that is used to build that brick wall are these little green dots in here. You can see them, that you form chains that connect the NAMs and the NAGs. And those are amino acids. All right, so you've got, I think, 15 or so amino acids connecting each one of those together. And so, and then there's cross connections as well in here. So this whole thing is held together very tightly, sort of like a chain link fence. And so, yeah, you've got multiple layers of this too. So here's one layer in here, then here's a second layer in here, and there's a third layer. And it's probably even going to be thicker, sheets upon sheets upon sheets of this meshwork of NAMs and NAGs and amino acids. It's going to be thicker, of course, in a gram positive than in a gram negative, but they both have it. Now, if you can introduce some chemical that will disrupt the synthesis of this, in particular something that disrupts an enzyme that maybe is active in helping to join the NAMs and the AGs via these amino acid linkages, you can promote a weakness in the cell wall that will lead to lysis. And that's what's shown here. You notice this is a compromised cell wall. It's got the NAMs and the NAGs, but look at these chains of amino acids. They're incomplete. And so those layers upon layers of NAMs and NAGs are going to basically just be shed. They're going to be jettisoned. They're going to fall away. And that cell is going to be really flimsy and weak. And so here, this shows a coccus type of cell, a staphylococcus, for example, with its peptidoglycan intact. But if in the process of that cell developing, growing, it is subject to treatment with cephalosporin or penicillin, you wind up with a cell wall like this. And here's the end result. So it's going to take on water and expand, and eventually it's going to go through lysis. All right. So yeah, this shows up here, this is a picture of it's an electron micrograph. You can see there's like a little filter back here, and they're collected on that filter. See, there's actually some of these in the process of dividing. Little splits between them. Anyway, those are properly shaped. it can be assumed they are viable staphylococcal cells. And here's what happens with them. This is another picture of some that have been treated with either a penicillin or a cephalosporin. And you can see all this junk in here. Well, that's residue. That's debris from cells that have broken apart. And you can see they're not shaped properly. They're kind of rough looking down here. That indicates that they're starting to fail. All right. So that drug is effective, at least in this instance, in destroying those staphylococci. And could be effective against some other bacteria. It just so happens that in this instance, that's what they were studying. So, these drugs that act on the cell wall, penicillins and cephalosporins, are effective because they contain just one little sort of functional chemical group, and it's called a beta-lactam group. And we'll look at that here in a second, what it looks like. But it contains 3 carbons and 1 nitrogen, right? All bound into this little kind of geometric structure. Looks like a little square, essentially. But that's sort of a signature chemical marker of one of these drugs. It's the beta-lactam group. Beta-lactam moiety or something like that. I forget what chemists call it, but it's the main marker of these. All right, and so that gives them the capacity to disrupt the enzymes that are involved in cell wall synthesis. That right there. All right. So the rest of the drug is sort of a vehicle for that, essentially. The rest of the molecule. The molecules are bigger than just that beta-lactam, as we'll see. And as it says here, greater than 1/2 of all antimicrobials are beta-lactams. It's actually like 70% of them are beta-lactams. That includes the cephalosporins and the penicillins, as well as some others. Those are the main ones. And so here then is the beta-lactam component, right? These are molecular structures of these things. And you can see that they all have, every last one of them, all of these different, in this case, psilins here. So these are all cousins of penicillin. You can see psilin in their names. Right? And that's a common theme. We would immediately know that there's beta-lactam in their chemical structures. There's also this other component here called a thiazolidine. And anytime you see thiazole, that refers to something that has both sulfur and nitrogen. So there's this nitrogen right here, and there's this sulfur right here. So those are thiazolidines. They too are sort of complexed with the beta-lactam, all right? And because that too is also a signature chemical, this is referred to as the nucleus of the drug, all right? So we've got nuclei of cells, and that's a different thing altogether. Here we're talking about a nucleus of a drug, and that is this chemical sort of center, if you will, that they all have, all right? So any penicillin is going to have that. And so different ones of these that are out there are unique because of the R group that they have. So R groups, assuming that stands for radical group. I've never known that for sure, but that's what they are, R groups. These are like the fingerprints that make each one of these unique. Right? So nafcillin has its R group, which is a couple of benzene rings up here, 2 6-membered rings. And then ticarcillin has this 5-membered structure that has a sulfur in it, and so on and so forth. You can see a lot of variation there. And each one of those is going to be produced by a different fungus. I'm assuming that these are all made by fungi uh, via fermentation. Okay, so um, there's that. And then, you can also make some semi-synthetic forms utilizing just straight up penicillin and in the laboratory add new things to it. So that's possible as well. All right, so yeah, here are the three parts: thiazolidine, the beta-lactam, and this variable side group, this R group, is what makes each one of those different. So if you got, say, 70 or 80 different ones of these, they're all going to be different with regard to their R groups. But it's that beta-lactam that's kind of like the key to them being able to disrupt cell wall synthesis, right? And then there are the cephalosporins, which are likewise mainly products of fungal um, uh, activity. They can be synthetically altered. Right? Many of these are products of particular organisms, though, and they account for 1/3 of all antibiotics. Right? And so you can see the beta lactam is here as well in the nucleus, but it's got a little different um, uh, partner here in the nucleus. It's not a thiazolidine, but it's another sulfur-containing structure. Right? And so you can tag onto this R groups that come off the beta lactam. All of these here are coming off the beta lactam. Or you can add R groups on to the other member of the pair of things in the nucleus. So you got even more ornamentation, if you will. You can add different little side groups and come up with different combinations. And quite a number of these are, in fact, semi-synthetics at least. All right. So now, one thing about them is this is not as desirable as one would like. Many of them are relatively broad spectrum. Um, they are, but many of them, though this could have changed the since the publication of this information. Um, Many of them are resistant to penicillinases. Now, penicillinases, let's talk about those real quick. Penicillinase, what is that? This is an enzyme that works against a penicillin or a cephalosporin, right? And these are the chemicals that are produced, and there's a genetic basis to this. So if an organism has the gene for making one of these penicillinases, it's going to be resistant to cephalosporins or penicillins, right? So these are the weapons that bacteria utilize to fight off the drug, all right? So when we talk about antibiotic-resistant forms that are resistant to penicillin, this is what they have, all right? This is genetically coded. So it's a weapon against a weapon, essentially. It's what they fight back with. So if you get an infection with, say, Streptococcus pyogenes, and it causes a strep throat, and your doctor administers a penicillin, and it doesn't work, it's because that particular strain of Streptococcus pyogenes is making penicillinases that are effective against that drug. So you're not going to kill it with that antibiotic. You're going to have to take something else. So what this is saying here is that there are a number of cephalosporins out there to which a lot of organisms have not evolved, have not adapted by producing penicillinases that are effective against them. So that's, there's both good and bad with that. If they're broad spectrum, we don't really want to take broad spectrum drugs, but they're resistant to penicillinases, so they might help deal with an infection that would otherwise be intractable. And then another thing about them is that they tend to be low in terms of their allergenic properties. So they're not going to trigger allergies in the infected person, which is something we don't want to do either. All right, so, and it talks about how they're administered. Okay, so you can administer many of these orally, which is for some folks what they prefer. They don't want to have an infection. But parenterally, this means to be introduced, through hypodermic injection, but maybe some other approach that doesn't involve ingesting it, so taking it. So parenteral means outside of the gut, right? You have to administer it that way. And with the cephalosporins, their names commonly contain roots like this. So you can see in the names of these things over here, you got the CEFs and CEPH roots. Moxolactam, that one kind of goes against this, but you can see lactam in there. So that's referring to the beta-lactam group. And you might look at that and say, oh, that could be penicillin. You'd be close. Either way, it's actually a cephalosporin. So anyway, there's what we call different generations of these now. You can see there's a first generation, a second generation, a third generation, fourth generation. These are, every time they produce a new one to counteract cephalosporin-resistant organisms, we say we're making a new generation of the drug, right? So, new improved versions are new generations. Now, there are some drugs that can inhibit cell walls that are not considered to be part of the lineage of penicillins or the lineage of cephalosporins. They're not really all that closely related to them. But one is vancomycin, which is a very powerful drug. But in recent years, Staphylococcus, Staph aureus in particular, infectious Staph aureus serotypes have evolved resistance to vancomycin. And so we have this strain of Staph aureus known as Versa now. Used to be vancomycin, but before the evolution of Versa, or the appearance of that, vancomycin was regarded as a drug to kind of not overuse, keep it on hand as a drug of last resort. If everything else fails, let's pull out the vancomycin and that'll take care of it. But now, of course, that's not even possible with certain of these strains of Staph aureus. But it's got the narrow spectrum property that's desirable, but it is toxic and hard to administer, so its use is restricted. Then we got bacitracin. That's a product of Bacillus subtilis, which we've actually worked with in the lab. That was before all of this hit. We were growing up some Bacillus subtilis and utilizing that to study endospores and other aspects of bacterial structure. So yeah, bacitracin is an antibiotic that's produced by Bacillus subtilis. It's a common component of ointments like salves, like I think Neosporin and some other kinds of antibiotic ointments, which can be applied to cuts and things on your fingers and so forth. And then isoniazid, INH, and this is something that can be utilized to deal with the tubercle bacillus, Mycobacterium tuberculosis. So this one is a cell wall inhibitor, but of a different sort. This is one that inhibits mycolic acid synthesis. So it's going to be obviously narrowly specific for those particular kinds of organisms, the mycobacteria, including leprosy and tuberculosis, as well as nocardia, those forms that can cause nocardiosis. And then we have drugs that can disrupt cell membrane function. All right, so um, we're going now in past the cell wall. Of course, this is going to mean that they're going to be a little more toxic because their cell membranes are kind of like ours. So if we take these drugs, going to probably do some more damage. All right. So, but the rationale behind them is if you damage the cell membrane, you disrupt metabolism of the organism, obviously, and you could lead to lysis as well. It could lead to lysis as well. And one way to make them specific for a particular microbial group is to alter lipids that are found in them. These are lipid-containing drugs. And so they tend also to enter into cell membranes, which contain lipid. All right. These are nonpolar drugs. Cell membranes tend to be nonpolar, and so these drugs will enter into them. Some of them act as surfactants. So yeah, your polymyxins. And then these drugs down here are primarily utilized against fungal infections, amphotericin B and nystatin. They complex with the sterols, things that are cholesterol-like, in the fungal cell membrane. And so we have some of these drugs that are utilized for dealing with fungal infection as well. So this shows what polymyxin B does. Next, essentially as a surfactant. And you can see it's characterized here by this little ring with a little side group hanging off of it. I don't know that that's what it really truly looks like, but anyway, you can see it's going in and complexing with the phospholipids in the phospholipid bilayer, and ultimately that leads to the disintegration of that. All right. See, it's interacting with the phospholipids, not these proteins in here, which tend to be polar hydrophilic things. The phospholipids themselves are nonpolar for the most part, and that's where the polymyxins go. And then you got drugs that affect nucleic acid synthesis, right? So they can block the synthesis of nucleotides, so adenine, cytosine, guanine, thymine, uracil. You can block the synthesis of those so that you don't have the raw materials from which nucleic acids, DNA, and RNA can be built. You can inhibit replication, so that stops the copying of DNA. And then you can stop transcription, which stops the production of RNA. If any one of those processes or all of them is disrupted, obviously that's very dire for that particular cell, because the DNA and the RNA are where all of the control of that cell resides. All right, the cell's going to lose the ability to control its function. And so recently, chloroquine, hydroxychloroquine, and other drugs have been in the news as a potential agent to deal with viral infection. And they work principally by binding and cross-linking the DNA double helix. All right, so you can take a double helix, and normally it would look like this. There's one side of it, and here's the other side. So it's like a ladder that's been twisted, right? And so what cross-linking entails is taking two sections of this and building, strong bonds between them. like bonds that run this way in here. And that's going to stiffen this up, and it's going to prevent any sort of, say, enzyme. Say this is a big enzyme that moves along this and reads it off. When they hit places like this, they're stopped, they're blocked. All right. So that's what, very crudely, very informally, that's what cross-linking does. And so chloroquine and other related drugs disrupt that. Drugs called the quinolins inhibit DNA helicases. DNA helicases basically are enzymes that take an intact double helix and open it up. All right, so you got a DNA molecule where there's bonds between the two sides. A helicase will come along and open this up. All right. So those open places in there are places where new nucleotides can be brought in to build now two pieces of DNA. All right. So the helicase is what gets that process started. That's what unwinds the DNA, if you will. It unwinds the DNA. So if you can disrupt that, you're going to shut down replication. That's what the quinolins do. And then you've got some drugs. Now, this does mention some antiviral drugs besides antibiotics, but some of the antiviral drugs work because they are analogs of purines and pyrimidines. There's one called HZT, or AZT rather, an antiviral drug that stands for azithromycin. So the T in there indicates that this is related to thymine. So it's a thymine analog, which means it's not exactly thymine, but it's a lookalike. It's a doppelganger. And so if you introduce this into a person who's infected with a virus in particular, as the virus sets about replicating, it will take the AZT and build it into its genome in places where normally a thymine would go. And that disrupts it, right? That once that happens, that genome is now corrupted and it no longer will function as it should, all right? The problem with that is if you take AZT, it'll shut down the virus, but it also will be incorporated into your own DNA, and it will no longer function as it should in your body. Your DNA won't. And so it's another example of how we've got to do this cost-benefit, analysis and decide, is the risk worth taking? So if somebody's really healthy, they might be able to handle that. If they're already very sick, AZT may actually kill them. All right, it may take them past the point where they can continue. So that's how some of those drugs work, some of these antivirals that are analogs of purines. Chloroquine and quinolones are not analogs of purines, so I want to make that disclaimer. They do other things. All right, and then we have drugs that can disrupt the ribosomes and their functions, which, like I said, seems like a great target because they're different from our ribosomes. But you got to remember, your mitochondria are prokaryotic, and they have 70 S ribosomes, and these drugs will attack those. And so mitochondria are your powerhouses, and it's where ATP is being formed. So you can expect to have some pretty severe consequences of these drugs. The hope is that they'll have their effect fairly quickly, kill off whatever infectious agent is giving you problems, and then you'll metabolize the drug and secrete it and get rid of it, excrete it, and get rid of it, and be none the worse for wear. But oftentimes you'll come away with some problems as a result of having taken some of these. So the aminoglycosides, the tetracyclines, and chloramphenicol You can see that they target different aspects of ribosome structure. So aminoglycosides insert on the 30S subunit, which, when messenger RNA comes in for translation, that's disrupted. So protein synthesis is shut down there. Tetracyclines block transfer RNA from coming in. Transfer RNAs carry amino acids into the site of protein synthesis, and so there's an acceptor site on the ribosome known as the A acceptor site, and transfer RNA can't get to it. So that'll shut down protein synthesis. And then chloramphenicol shuts down peptide bond formation between amino acids. So, that's everything goes as planned up until that point. The ribosomes form, messenger RNA attaches, it gets pulled through, transfer RNAs are brought in to bring amino acids. But, when the amino acids are then finally to be brought together to make a real protein, that can't happen in the presence of chloramphenicol. So the infectious agent doesn't get the proteins that it needs to keep it going, so that's going to shut it down. Unfortunately, that's something that's going on in your body as well. You're making proteins, and if you've got chloramphenicol in place, you can't make your own proteins. You might make some of them, but not enough of them. So that's why that's so dangerous. So some of these are kind of dangerous, right? Got to really think about when to use them. Got to be appropriate context within which they're used. So yeah, this shows all of the different possible targets of the 70S ribosome. These little symbols here stand for the different drugs and where they go. The how they get involved in disrupting this whole thing. So all of these are things that disrupt the process, in this case, of translation. It's disrupting translation of genetic information. All right, so we've got all of those, which are principally antibiotics or semisynthetic forms of antibiotics that have been modified later. And I even mentioned a few antiviral and antifungal things in there. But there are those things, and then we've got, as a last sort of vulnerability, the metabolic pathways of infectious agents, of bacteria in particular. And so one of those that is specific to bacteria that we don't have, right? This makes this a fairly narrow spectrum approach. Is this right here? This is the metabolic pathway that we're talking about. So this is specific to bacteria. We don't have this. This doesn't occur in our cells. If we disrupt this pathway, you can see it's got these different components in the pathway. I'll just give them little letters here. So those are intermediates in a pathway that has basically really 3 steps in it. 1, 2, 3 steps. All right. And so what they're doing is this stuff, PABA right here, that stands for para-aminobenzoic acid. I don't know if I should have. Looking for it written down here somewhere. Oh, anyway, aminobenzoic acid. That's PABA, right? And that's a substance that bacteria can use. Right? We don't use it. They take it up as a substrate. There's an enzyme number 1 here, whatever that is, acts on PABA to turn it into this stuff here, B, dihydropteroic acid. P is silent in that, by the way. Dihydropteroic acid. Now, that is an intermediate. The bacterium doesn't use that directly, but it can convert it into to this stuff here, dihydrofolic acid, right? Or it's sometimes called dihydrofolate, okay? And enzyme 2 catalyzes that conversion. And then dihydrofolate, or dihydrofolic acid, C, is converted with enzyme 3 here to the final product, D, which, lo and behold, is folic acid, right? This is a vitamin. This is one of the B vitamins. And you know it's really important for you. Now, we get it from the foods that we eat. We don't actually synthesize this. So this is one of the essential nutrients that we would need. We get it from, well, as the name suggests, from foliage. You can get it from green leafy vegetables and other sources. These bacteria have to make their folic acid, right? They need it too, but they make it. And so this is the process by which that is done. Now, I'll tell you, if they don't have folic acid, they don't function. It kills them because they need it to make all of these things. Purines, which are adenine and guanine, they need those for their DNA, right? And their RNA. Pyrimidines are cytosine, thymine, and uracil. They need those. They don't have those, hey, they're out of luck. And then amino acids are used for making proteins. By gosh, you better have folic acid, which is involved in the processing of those things, synthesis of those. So they make their own folic acid. Well, guess what? These drugs, known as the sulfa drugs, commonly, these are sulfa drugs, right? These are the ones that were utilized prior to World War II. When penicillin was being developed, we did have access to these. So if a soldier on the battlefield was hit with a round, it was bleeding and had a wound, they would literally take a powder containing these sulfa drugs and just dump it onto that wound in hopes that the bugs, the infectious agents, wouldn't cause infection. That was one way it could be administered. But yeah, sulfonamides and trimethoprim are sulfur drugs. These are synthetics, and they block right here. So that enzyme number 1, this will bind to that enzyme. The sulfonamides will bind to that enzyme and shut this process down right there. And then trimethoprim blocks there. So take both of those. You can take one or the other. If you take both of them, it's like hedging your bets. That's added insurance that you're going to shut down this process, right? So the idea is if the sulfonamide didn't work to shut down enzyme number 1, well, the trimethoprim will back it up and shut down enzyme number 3. Taking both of them together yields what's called a synergistic effect. And there are some drugs that are of this variety that are still taken. It's actually very common for children who get frequent ear infections and so forth to be administered to be treated with these drugs. They can cause some allergy sometimes, so once that's determined, it's they're kind of off the table as options. But there's one drug called Bactrim, which contains these and some others that are commonly used. Anyway, this is how they work. But when we say they inhibit enzymes, this is exactly how they do it. Here's an enzyme down here, this little green blob, big sort of wad of protein, and on their surfaces they have these active sites. Here's the active site, and normally PABA, which has a shape that matches that site, will fit in there like a key in a lock. And the enzyme will alter it and turn it into substance B here, all right? And the process can go forward. Well, what the sulfa drug does, in this case the sulfonamide, what it does is it has a shape that's sort of like the PABA. At least one aspect of it is. And if you've got enough of this stuff floating around, hey, it's going to take up residence. It's going to just enter into and bind to the active site ahead of the substrate. And the thing about this is these sulfa drugs are very, very likely to bind tightly to those sites. So the substrate, the PABA, can't get in there. So that's going to shut that down. That's how they work. Okay. So that's a form of inhibition. That's called competitive inhibition. They're competing for the same site. All right, competitive inhibition. That's what we got going on here. There are, by the way, inhibitors that are non-competitive, and what they do is they bind to other parts of enzymes besides active sites. Okay, both of these sulfa drugs and trimethoprim are competitive inhibitors. They bind to active sites. So non-competitive inhibitors are things that are really tenacious and really effective at blocking enzymes. Unfortunately, there aren't any of these drugs that are like that. These are all competitive inhibitors, but they work well enough. All right, so yeah, you can see this shows a more detailed look at the PABA, which is the bacterial substrate. That's what it usually uses to make folate. So it's ultimately going to be converted into folic acid. And then this is the drug that's the inhibitor. All right, that's the sulfa drug. That's a competitive inhibitor that we would take on purpose to shut this process down, to basically treat the infection. So aminoglycosides are antibacterial drugs. They don't target peptidoglycan synthesis. That would be your beta-lactams, your cephalosporins, and your penicillins. These don't target the cell membrane. It comes down to protein synthesis and DNA replication. It's, I believe, DNA replication. Back to that real quick. Yeah, here's your aminoglycosides. Actually, it's protein synthesis. I'm sorry. Blocking there. Anyway, I'm not going to ask really specific questions about that on this exam. But yeah, that's the answer right there. Protein synthesis. All right. I'm going to keep going with this. So let's talk about this topic. This is one that sometimes we don't get to in the regular term, but the acquisition of drug resistance, antibiotic resistance. That's what this is about. A fairly basic concept. It's not super challenging to understand, but there are some misconceptions about how it works, and those are especially annoying to me sometimes when I hear people discuss this and they have it all wrong, and they assume that that's how things work, and they don't. So, well, what this is, it's an adaptive response, right, that organisms have where they are able to tolerate a drug that would normally kill them. Right? They become resistant to something that formerly they were sensitive to, that they would have been inhibited by or even destroyed by. And they are able to do this. They are able to adapt, not because they get stronger. It's not like you can take an individual bacterial cell that currently is kind of weak and and unable to resist these drugs. Not like you can give them a little bit at a time and make them stronger, right? It's not like that. It's not something that individual cells can get stronger in their abilities to do. It's a population level phenomenon, right? It involves reduction in certain genotypes, an increase in others. So a genotype is a genetic variant, right? Kind of like in a human population. Some of us have brown eyes, some of us have blue eyes, some of us have green eyes, and whatever other eye colors there are. And those are genetically determined, all right? So, God forbid, let's say all the brown-eyed people were to suddenly be wiped out by something that they're sensitive to, some chemical in the environment. They're unable to survive or reproduce. Brown eyes are going to go down in frequency, and the other eye colors are going to become more common. All right. So it's not like individual cells getting tougher and stronger. It's like populations completely shifting their makeup so that the population comes to be made up of more strong individuals than was the case previously. Right? It's differential survival among these different genotypes is what it is. And so, yeah, it has to involve genetic versatility, genetic variation, the ability to meet these problems is a consequence of the population's inherent genetic variability. So if you had a population that was composed of 100%, everybody in that population is completely sensitive to a particular drug. There's no variation. Everybody's exactly the same. You hit it with that drug, it's going to kill them all. All right. If you have, on the other hand, a population that has some that are resistant, say 95% of the population is susceptible to the drug, but 5% are resistant, and you hit that population with this drug, it kills off most of them, but it leaves behind that small number, that 5%, which are survivors that are genetically able to handle this drug. Guess what they're going to do? You know how bacteria are. They have very short generation times. They'll quickly go through generation after generation of binary fission. They'll reproduce like crazy. They'll build their numbers right back up to where they were before the drug. Guess what? Now the ratio of susceptible to resistant individuals is biased toward resistant individuals. 95% of them are resistant, and 5% are susceptible. So that's what it is, the shift in the makeup of a population. That's what it is, all right? And so there are forms of it that are intrinsic. So you have, let's say you have an organism that has a gene for resistance to penicillin. It already has it. Penicillin's never been introduced into the surroundings. It's never encountered that, but it already has this gene in place. It didn't have to get it from anywhere else. It already has it. That's intrinsic resistance. Now, if you have an organism that currently is not resistant because it doesn't have the gene, guess what? There are ways for it to get that gene, all right? Some very simple ways for it to get that gene. So that's acquired resistance, all right? And that's what makes bacteria so insidious, is because they have that ability, all right? They're able to acquire their resistance. Even though they currently don't have it, they can acquire it, and we'll talk about how that happens, all right? And in both cases, we're talking about genes. You either have a gene to start with, which is intrinsic resistance, or you pick up a gene and become different as a result of that. It's acquired resistance. All right. Let's talk about that. How does that happen? How do you get resistance when you didn't have it to begin with? Well, there's three paths, and here they're indicated in this diagram. So this one is called transformation. All right. This one is called conjugation, and this path is called transduction. So we got transformation, we've got conjugation, and we got transduction. 1, 2, 3 different paths to acquiring resistance to a drug. So transformation, let's look at that picture. So here we've got an organism, and it's got DNA fragments, right, that are being released as it dies. So this bacterium here is just basically falling apart. It's one that's lived out its life, or it's succumbing to some other damage. So the source of the genetic material, this guy right here, this is the recipient. Let's call him the recipient. This is the one that's acquiring the resistance. This guy can actually literally take up DNAs from outside sources that are being released, in this case, by somebody that's dying. And here's the thing. This source here, the guy that's dying and losing its DNA and it's going out into the surroundings, it doesn't even have to be the same species as the recipient. Okay, so let that sink in. So the recipient here, let's say it's E. coli, just for sake of discussion. Let's say it's E. coli. This guy could be somebody else. This could be some bacillus species, maybe. Maybe that's not the best choice, but let's say that it is. So it could pick up a gene from that. Now, among the mix of DNAs that are coming from the dead guy, there might be one of them that's a resistance gene, right? And it gets taken up by the recipient. And all of a sudden, boom, this guy now, recipient, has that resistance gene. And so if it's resistant to penicillin, if that confers resistance to penicillin, this new recipient is not going to be affected by it. Whereas prior to the transformation, it might have been susceptible to penicillin. So, this is very scary. Live bacteria can pick up genes from dead bacteria. Pick up DNAs from just random sources. And, now are changed. Note that this is not a reproductive process. This is simply that one cell, that recipient, undergoing some change. You still only have one cell when this is all done, but that cell is new. In a genetic sense, it's new. That's path number 1, transformation. Path number 2 is conjugation. So up here, this is our new source in this picture. And I apologize for the horrible handwriting with my finger. But anyway, you can see the source in this instance has a plasmid, right? A little circle of DNA. And part or all of that can be transferred by way of a pilus. Now, it doesn't show the pilus here, but remember, a pilus is a little short, hollow, stubby little pipe through which things pass from one bacterium to another. So this guy here, the source, if it has a resistance gene as part of its plasmid and it sends that on into the receiving gene that didn't have the resistance, the receiving bacterium, that didn't have the resistance gene, hey, guess what? It picked it up. So conjugation is transfer from one live bacterium to another. Right? Transformation is passage of genetic information from a dead bacterium to a live one. Right? And then the final path is through viral transduction. So here we've got a bacteriophage. It was assembled inside of a host cell. It has its own viral DNA, but guess what? It could have picked up some DNA from the bacterium within which it grew. So it could be a mixture of viral and bacterial DNA that it has. And if one of the genes that the virus picked up while it was being assembled in the bacterium is a gene for resistance, and that guy then leaves the host cell and finds a new host to infect, it can shoot its resistance gene in that way. It can shoot its genetic material in containing the resistance gene, and that now can be incorporated into the genome of the host, right? Because remember, sometimes bacteriophages do what? They've got two cycles. They got the lytic cycle and the lysogenic cycle. So if this goes in here and becomes a prophage, if there's lysogeny that happens, then this bacterium is going to be able to live, even though it's been virally infected. It'll be able to live, and it'll be resistant to an antibiotic. Okay, so those are all different paths through which antibiotic resistance can be acquired, right? Now, if this recipient cell already had a gene for resistance and it hadn't had to acquire it through any of these pathways, that would be an example of intrinsic resistance. But these are all acquired forms of it. Another way to get resistance is simply through mutation of your own genetic material. So that can happen too. So mutations happen. Sometimes those lead to development of new genetic variants, new alleles that give one the ability to resist antibiotics. So that's a fourth way. By the way, this term here, transposon, that refers to a piece of genetic material that's incorporated into a chromosome, that a gene essentially that can move from one place to another in the genome. Those are sometimes called jumping genes, and it's theorized that they are these pieces of viral DNA. Originally, they were pieces of viral DNA that got incorporated into the genome of receiving organisms. And they can confer drug resistance on those that pick them up. Okay, transformation, conjugation, and transduction. So remember those. And just spontaneous mutations. That's another way that you can acquire resistance. And so when you get the resistance, when you get the gene for resistance, when it is expressed, what does that lead to? What are the mechanisms of resistance? All right. Well, one I've already talked about is penicillinase. So if you pick up a gene that lets you produce penicillinase, which is an enzyme, you can then take that enzyme and use it as a sort of pair of scissors to chemically cut into the penicillin. So here's our beta-lactam ring. Well, in the presence of penicillinase, that ring gets opened up right here. And that no longer is functional. That's not going to disrupt peptidoglycan synthesis. The bacterium can go on its merry way. So the penicillinase is its weapon against penicillin. All right. So this is something that a drug-resistant and antibiotic-resistant bacterium would have. More than likely, if it's resistant to penicillin, then that's what it's using to be resistant, to confer that resistance. And then you can see these are other ways that bacteria can acquire resistance. Right? So they can normally drugs, when they enter, they bind to receptors in the bacterial cell wall or cell membrane, and they're brought in that way. Well, you might pick up a gene that causes your receptors to change shape, and they no longer bind the antibiotic. So the antibiotic, in order for it to be effective, has to go into the cell. You can see here it's blocked. There's no way it's getting in. All right, so that's another means by which resistance actually happens. Okay? You can acquire the gene for this, and it'll do that. Drug pumps. Okay, so gram negatives often have in their outer membranes pump proteins that pump things into and out of the cell. Sometimes a drug is mistakenly pumped into the cell and it will kill the bacterium. Well, if you acquire the right gene, it can change your drug pump, your pumps, and those pumps are made either incapable of pulling in the material, or they'll actually act in such a way as to pump the drug back out. Okay, so that's another way to go, activating drug pumps. And then we just talked about aminoglycosides operate on protein synthesis by binding to ribosomes. Some drugs can prevent those drugs from attaching to the ribosome. So some resistance mechanisms, operate by blocking the drug's attachment to the ribosome. Right. So here you get a piece of, in these two cases here, you've got in a resistant bacterium, you've got ribosomes that have bound messenger RNA and transfer RNA, and they're about to go ahead on and produce new protein, and that bacterium is going to be happy. Even though the antibiotic is present, the antibiotic is rendered useless. It's not going to do its job. And then, sulfa drugs, trimethoprim and others, those can be blocked by bacteria that evolve ways to counteract them. Okay? They can pick up a gene. If you're an organism that's normally affected by sulfa drugs, you pick up a gene through transformation or conjugation or through transduction or through spontaneous mutation, you might not be susceptible to sulfa drugs anymore. You might wind up being resistant to them, and they would be ineffective. So those are all possible things. Yeah, this is what I was talking about earlier. So it's not like individual cells develop immunity. They don't have immune systems. They don't have antibodies and things inside of them. They're single cells, for gosh sake. So they can't do that. But instead, what happens is the whole population changes. So here's a plate down here containing microbes that are plated out there. And just by random, you can expect through spontaneous mutation, even before a drug has ever been presented to a population, there's probably going to be a few cells in there out of the many hundreds of billions, if not trillions of cells. There's going to be a few of them that are resistant to the drug. Normally their numbers are really low because there's no reason for them to be all that common. But if you then expose that population to the drug, you wipe out most of these resistant guys, boom, boom, they all go away. And who do you have left? Those two little guys there that were off by their lonesome, that were all lonely, and there were hardly any of them. Guess what? Conditions are such now that they can reproduce. They're the survivors, and this is what you get. A population now that, after exposure to the drug, shifted in its makeup over to this other type, right? So there had to be a lot of death in order for this to happen. You wind up with a lot of dead cells. The survivors go on to replace those that died. That, my friends, is natural selection. That's how it works. This population has evolved. This is evolution in action. So if somebody says, well, evolution, I don't believe in that, it doesn't ever happen, well, here it is. This is what's going on. This is why we are running out of antibiotics that are effective. They're evolving resistance to it, right? And it is natural selection. The reason we don't see it in larger organisms as frequently is because their generation times are much longer. So it happens in larger organisms, but at a slower rate. So by the time meaningful results of natural selection occur in larger organisms whose generation times might be measured in decades, we'll already be dead. So we don't see natural selection happening as frequently in our lifetimes with larger organisms as we do when we look at these smaller microbes that can reproduce in what, like 15 minutes, right? So that's what it is. That's what's going on. And I can tell you, and I've said this, if you give me just say some natural occurring microbe, whatever it is, go to the restroom at Jackson State and swab the sink or some other place in there and plate out some organism, isolate that organism, get populations of it going. If we then start subjecting them to doses of antibiotics, say in the medium or in some other way, we'll ultimately be able to select from an original population resistant strains. We can actually do that fairly quickly in just a number of days, really. That's why when a doctor administers antibiotics, when he or she says, take all of this and take it according to the schedule that is on there, don't, quit taking it after you start to feel better and leave half the bottle of pills. That's why they're telling you that. You want to kill everything. You want this whole plate to be wiped out, right? If you don't do that, if you don't take your drugs properly, you'll wind up producing something like this. And that's why we are in the position we're in currently with antibiotics. We're losing those as weapons to help us in dealing with bacterial infection. Right now, we got a pandemic involving a virus. I'll just tell you, it's possible that in the future we could wind up with a similar thing involving bacteria. Right? So if you're sufficiently sort of on edge now, maybe you will be as a consequence of hearing that. I don't know. But that should go into that should inform this from here on out with regard to when you get sick and somebody asks, do you want an antibiotic for this? If they don't know exactly what you're sick with, if it could be a viral infection, you don't need to be taking antibiotics. And certainly if you're feeling just a little bit under the weather, but not really sick, antibiotic may not be what you want to take right at that moment. Because like I said, it could be a viral infection. I don't know. There's a lot of ways that this information could inform the decisions that you make in the future. Hopefully you do that. All right, so yeah, these are some of the things that can be done to help us limit drug resistance in the future. Doing these things and supporting these things, supporting drug research and other long-term strategies. Making sure that the people that you elect to office are folks that are aware of these things and take them seriously, so that they can put proper plans in place. Should epidemic disease arise in the future, or the potential for it is there, they take pains to ensure that is accounted for and we're prepared. All of those things. Vaccine development, all of those things. Now, there are, like I said, adverse effects with these things. It's going to happen. There are a variety of interactions that can occur between the host, that would be the infected individual, and the drug that they're taking. And it could be, that the effects are serious, possibly life-threatening, life-altering in various ways. So 5% of all people taking antimicrobials will experience that. So, yeah, it could be damage to tissues due to toxicity of the drug. So some drugs cause issues with your kidneys. So renal failure could be a result of some of them. Others do damage to sensory abilities, like hearing loss can, what's called ototoxicity, and attend the use of some of these drugs. Streptomycin is one that is known for that. And there's a number of that involves hearing. Get some very delicate little hair cells and other things there that are part of the inner ear hearing apparatus. Those can be damaged. So ototoxicity and other things too. I mean, other functions as well. So tissue damage, organ damage, allergic reactions are different. Obviously, these are things that when you take them are seen by your immune defense system as antigens, and the response then is to produce lots of histamines, cytokines, and have this explosion of those in the body. And that's an allergic reaction. That's anaphylaxis. And some of these can trigger that kind of response, which could be obviously life-threatening. So we want to avoid that. So that's your body's own response to the drug, which is seen as a foreign substance that it needs to respond to. It's not due to the drug's toxicity or anything like that. And then, of course, these drugs, when you take them, will kill the stuff you want to kill, but then could kill some things you don't want to kill. And one consequence of that ultimately is what's called a super infection. So you wind up encouraging the growth of something that's infectious and getting sick as a result of that. Here we got a person that has had some discoloration of their teeth due to having taken tetracycline at some point earlier in their life. So that's something that can happen. There's a few other antibiotics apparently that can do that as well. So that's not life-threatening or anything, but that's something that's permanent that isn't going to go away. So apparently in this person's youth, as their teeth were forming, there was some disruption of that led to this discoloration. So they're left with this permanent sort of record of that having happened. So here's kind of a diagram that shows what goes on in super infection. So here the patient has a sore throat. There's an infection there. Strep throat, Streptococcus pyogenes, or some other such organism that's infecting the pharynx. And so they take a drug, and the drug is going to course through the entire body. They can't really just target that one specific locality. And as a consequence of that, it makes its way into other compartments. There's your gut. This person's gut's functioning normally, but part of the flora that's there includes a few things whose numbers are really low that are potential pathogens. And among those would be C. diff, Clostridium difficile, which is a potentially very dangerous pathogen, one that has antibiotic resistance. There's other things too. I mean, you can have Enterococcus faecalis and other organisms that likewise are potential pathogens. They're living there in your gut, and their numbers are held in check by these other good bacteria that are part of the flora. Now, if you take the drug and it circulates, those good guys get knocked out. The resistant, potentially pathogenic forms survive, and they come to make up a greater proportion of the community. So this isn't a population level thing. This isn't really evolution. We're not talking about one population of one kind of organism with genetic variation. We're talking about a community of organisms, some of which are resistant, some of which are susceptible. So these little blue guys, that represents one species, and the little red triangles are another species. So one species is wiped out. The other one is now freed from inhibition, and it takes off. And look what happens. Hey, you got a gut infection now. Right, that's super infection. That's a second infection that results from having um, taken a drug which has altered your microbial flora in your gut. That's what that is. That's one of the possibilities when you take antibiotics, is that sort of thing happening. All right, so there's that. Okay, so I'm going to stop it there, obviously. I've got to stop there. And for now, that's my plan, is to just go with that as the material that we're covering for this last of your exams. So today is the last day for the exam that's up there right now. I think it goes until midnight tonight. So I'll be posting this, and I'll wait a day or two and then go ahead and post basically the last exam, the last electronic exam. And that will be what I'll use for your lecture grade. For the lab, I've got the information that's up there on MVIC testing, differential and selected media, and then the review of organisms. Those three things are up. They're posted. Review those, and here probably today I'm going to go ahead and post some quiz/exams on those, and that's what I'm going to use for the lab. All right. So anyway, I'm going to stop right there and be looking for those things coming along. So I think everything has to be done by May 5th. So yeah, we're approaching the end of all of this. So anyway, watching eLearn. The stuff will show up here shortly.